Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi ta'ala wabarakatuh, and welcome back everyone to UMC 463 Meet Your Microbes. Inshallah, today we'll be covering viruses, its characteristics, its classifications, and its importance. By the end of today's session, hopefully you should be able to describe the basic characteristics and classifications of viruses. In addition, it is hoped that you'll also be able to demonstrate your understanding in terms of the beneficial roles and clinical significance of viruses. Let us start with virus characteristics. So let's dive in. Viruses are tiny infectious agents that can only replicate inside host organisms. They may be tiny, but their impact is enormous. Now let us talk about its structure. Viruses are very simple beings. They're not prokaryotes like bacteria. They're also not eukaryotes like fungi. They are acellular organisms, which means they don't have any organelles. Instead, they have either a DNA or an RNA inside a protein coat, and that's it. In terms of size, they're very tiny. When we say bacteria are small, viruses are much more smaller. And one thing that is very unique about virus is that they are host dependent. They cannot do much without a host, which means no host, no viruses. One of the reasons why is that even the replication of a virus takes place inside a cell. In order for it to infect other cells, it has to be host dependent. And it affects all types of organisms, humans, animals, plants, bacteria, you name it, they'll infect it. The term virus actually comes from the Latin word which means poison. This signifies how dangerous and how harmful viruses can be when they get nasty. They're often referred to as obligate intracellular parasites. Now be careful, viruses are not parasites. However, the fact that they need the host more than the host needs them shows that they are parasitic in nature. Viruses cannot replicate on its own. In fact, it usually hijacks the host's metabolic machinery to do its business. And upon infection, a virus then directs the cell machinery to produce more and more virus for itself. So the host doesn't need a virus, but the virus definitely needs a host, which is why, as I mentioned just now, it is parasitic in nature. Although viruses are simple, as I mentioned previously, they only require nucleic acid and they only require protein, they are also very diverse. They're simple, yet they're complex in terms of its structure, in terms of its genome organization, and in terms of its host range. Viruses come in a lot of shapes and structures. You've got complex viruses in the form of bacteriophages, you've got helical viruses, you've got polyhedral viruses, you've got envelope viruses. So there's a wide variety of shapes for viruses compared to your typical cocci, bacilli, and comma-shaped bacteria. Irrespective of the size and shape, every single virus must have this basic structure comprising of nucleic acid, which is either DNA or RNA, it cannot be both, inside a protein matrix or a capsid. So that is fundamental. You've got your nucleic acid and you've got your protein, and that's it. Certain viruses, they may have envelope, but certain viruses, they don't. That's okay. And one thing that I want to emphasize is the nucleic acid, or the actual genome, is very vital because it's used for virus classification purposes. The virus genome could either be single-stranded or double-stranded. It could be linear or circular. It could also be positive sense or negative sense. And all of this will be used in the Baltimore classification system. So it looks kind of simple, but if you break it down, it's actually rather complex. But don't worry. Viruses are often confused with bacteria. So let us take a step back and explore the differences between bacteria and viruses. Firstly, the differences lies in its structure. Bacteria, they are living single-cell organism with a complete cell structure. And what do I mean by complete cell structure? It means that it's got a cell wall, cell membrane, it's got genetic material, it's a whole package. But viruses, on the other hand, they are non-living. They are essentially just genetic material that is wrapped up inside a protein. It's got no cellular structure and they cannot live independently. Secondly, the differences lies in its size. Both bacteria and viruses are microorganisms. Micro means small. But how small is small? Viruses are a lot smaller than bacteria. A typical bacteria size will range between 0.2 to 5 micrometers in comparison to viruses, which ranges between 20 to 300 nanometers. Even the units are different. For bacteria, it is measured in micrometers. For viruses, it's measured in nanometers. For viruses that is considered large, for example, cytomegalovirus or influenza virus or pox viruses, these are large viruses. But large is actually between 120 to 300 perhaps, max nanometers. So it's still not to the level of bacteria. Bacteria are small, correct, but viruses are much, much more tiny. To put things into perspective, this particular figure summarizes the comparative sizes of viruses versus bacteria. So in this figure, there is one bacilli. It could be E. coli, and you know E. coli are very, very small beings, but inside it, you can see diagrammatically, you can actually place lots and lots of viruses inside it. As I mentioned just now, viruses range between 20 to 300 nanometers, and if you can see that, smallpox is considered big. That's considered 300 nanometers. So if you can compare that to E. coli, you can understand that viruses are in fact really, really tiny organisms. And that's pox virus. Imagine if you're a polio virus. Imagine the size of a polio virus. It's about 20 nanometers. So that's how small is small. The third difference lies in the living status. Bacteria, as mentioned before, they're living organisms. Viruses are not considered living. Bacteria, they can live independently, happily, without a host. They can reproduce on its own. They can carry out metabolic processes on its own. They're very, very self-sufficient. In contrast, viruses are not. They need a host so that they can live. They need a host so that they can infect. The mode of reproduction is different for bacteria and viruses, whereby bacteria reproduce asexually through the process of binary fission, whereas viruses, they must infect a host in order for them to replicate. The laboratory diagnosis for bacteria and viruses are completely different. The lab techniques are different, and also the instrument used will differ. Bacteria, they can be cultured on various culture media. They usually grow very happily, providing that you give the appropriate nutrients, and you can observe the colonies with your naked eye. And if you stain the bacteria, for example, if you do gram stain or capsule stain or spore stain, you may require a microscope, but the microscope that you'll be using is a light microscope. For example, you do your gram stain, and using a light microscope, you can look at the colors. Gram-positive organisms will be purple, the gram-negative organisms will be pinky red. Light microscope will be sufficient. In contrast, viruses, you cannot look with your naked eye. You cannot observe macroscopically. Even Superman cannot do it. If you want to observe the structure or the effects, you would require a much higher magnification because they're very, very tiny. And you would, for this reason, need electron microscopy. Can you stain them? Yes, you can. But the stain you use will be different. And usually the output, the image created, will be different as well, because it's usually a black and white electron micrograph in comparison to a more vibrant and colorful images of bacteria. You can grow them in the lab, definitely you can. But you will not be using standard culture media because viruses requires a host. So if you want to grow the viruses, you would either need cell or tissue, and in that respect, you be using cell culture or tissue culture techniques. So the process, as I said, is different. The output is different. They're totally different diagnostic procedures for bacteria and viruses. And the last differences lies in its treatment. Bacterial infections can be treated with antibiotics, but antibiotics would not work with viruses. Instead, for viral infections, you would need antivirals. Next is virus classification. Traditionally, virus has been named based on multiple approaches. The first approach was naming a virus based on its host range. The second approach was naming a virus based on the disease they cause. And the third approach was naming a virus based on the place where the infection was first reported. For example, Newcastle disease virus, or NDV, was named as such because the first infection was in fact detected in Newcastle. Nevertheless, there are limitations to such approaches because certain viruses can infect multiple hosts and different viruses can cause similar diseases. Because of these limitations, modern virus classification systems now look at multiple criteria such as information pertaining to the genome type, the genome structure, the virion structure, replication strategy, and evolutionary relationships for naming a virus. When talking about virus classification, two things must come in mind. The first is ICTV, and the second is Baltimore classification. ICTV stands for International Committee on Taxonomy of Viruses, focuses on viral taxonomy and nomenclature, whereas Baltimore classification system emphasizes genome type and replication strategy as a method to classify viruses. So the International Committee on Taxonomy of Viruses, or ICTV, is the main authority for virus classification, and it uses a hierarchical structure similar to classifying other cellular organisms, including bacteria. So it starts with order, family, subfamily, genus, and species. The Baltimore classification system is a much more structured system that enables the classification of viruses based on their genome type and based on their replication strategies. This system divides viruses into 7 different classes. This is a summary of the Baltimore classification system, which categorizes viruses into 7 different groups. For UMC 463 Meet Your Microbes, you do not need to know in detail every single information pertaining to the description and examples per class. It is sufficient for you to understand that they are classified based on the genome type and the replication strategies. For example, Class 1 and Class 2 focuses on DNA viruses, but what differentiates Class 1 and Class 2 is that DNA viruses that is double-stranded belongs to Class 1, for example, herpes virus, and a single-stranded DNA viruses, for example, chicken anemia virus, belong to Class 2. Similarly, Group 3, 4, and 5 is pertaining to RNA genomes. What differentiates is whether it's single-stranded or double-stranded RNA and the replication strategies. So that's it for the first part of the lecture. Let's recap for a bit. Hopefully by now you should be able to appreciate the fact that viruses are acellular. They need a host. They must contain either DNA or RNA inside a protein. They're much more smaller than bacteria, and they are not independent in any way or form. And in terms of classification, you can either use ICTV, which is based on hierarchy, or you can use the Baltimore classification system to categorize the viruses into 7 different groups. So now hopefully you are ready to meet the good guys and the bad guys of the viral world. So despite meaning poison, not all viruses are bad. Let's start by looking at some of the good points of viruses. So, not all viruses are villains. They play quite a lot of beneficial roles in medicine and biotechnology, in agriculture, in scientific research, and in ecosystem maintenance. Here's the list of some of the good sides of viruses. Viruses play a very important role in medicine and biotechnology. For example, certain viruses have been utilized in gene therapy to treat patients. Secondly, for vaccine development, certain weakened and harmless viruses, such as vaccinia, have been used to make vaccines for therapeutic purposes. Third, for agricultural protection, certain viruses infect crop pests. So, this provides an alternative or reduces the need to use chemical insecticides, which can harm the ecosystem and can harm the crop itself. Fourth, is widely used for scientific research because viruses help scientists to understand the fundamentals of cell biology, the fundamentals of genetics, and how immune system works. Viruses also play a role in marine ecosystem balance because marine viruses regulate plankton populations and this supports the ocean food webs. And lastly, viruses play a role in potential cancer therapy because oncolytic viruses have been used and have been targeting and destroying cancer cells without harming normal ones. So this is good news for medicine breakthrough. Of course, many viruses act as pathogens, so let us look at some of the clinical significance of viruses. The trouble with viruses is that it is vicious. It causes infections not only in humans, but also in animals and also in plants as well. And in healthy individuals, usually what happens is it can remain dormant, but once your immune system is impaired or becomes weak, it can attack and cause opportunistic infections. So viruses, they are nasty. They're nasty because they can cause quite a lot of infections, and these are just some of the examples. You've got your influenza viruses, para-influenza viruses, H1N1, SARS, respiratory syncytial viruses, coronaviruses, including COVID, adeno and rhinoviruses that can cause respiratory tract infections. You've got rotavirus, norovirus, and enteroviruses that can cause gastrointestinal tract infections. You've got your HIV virus, you've got your hepatitis viruses, you've got cytomegaloviruses, which are blood-borne. Then you've got cytomegalovirus or CMV, rubella virus, varicella virus, parvovirus, and enteroviruses that can cause congenital infections, which means when a pregnant mother gets infected, they can pass the virus to the baby with detrimental effects. You got herpes virus, Coxsackie virus that causes skin infections. You've got avian influenza, Ebola, and hantaviruses, which are zoonotic in nature. And you got viruses such as Zika, dengue, Japanese encephalitis, which are vector-borne. And viral infections, they can either be symptomatic, which means you can actually look at the symptoms such as warts, genital herpes, pox, you can actually see it, or they can become asymptomatic, and this is actually quite dangerous because a person may feel that he or she is okay, but in fact they are chronic carriers. They carry the virus in the body, which remains dormant to a certain extent. Later on, they can actually get reactivated and cause harm later in life. So beyond diseases, viruses can cause a wide range of other negative impacts, including negative impacts towards agriculture, because plant viruses can cause reduced food supply and loss of income. Secondly, it can cause negative impact in terms of livestock, whereby animal viruses can kill or reduce the productivity in livestock, affecting the food industry and causing economic loss. Then this economic burden can also be seen in healthcare, it could also be seen in trade restriction, and it can also be a result of reduced productivity in general. Viruses can cause biodiversity threats because viruses can wipe out vulnerable wildlife population and disrupting the ecosystem. Viruses has the potential of being a bioweapon because viruses can be modified for harmful purposes, including for the purpose of bioterrorism, and that's kind of scary. And lastly, viruses can be used for public panic and social disruption because outbreaks can cause fear, outbreaks can cause social instability. So, in summary, virus could either be your best friend or it could be your number one enemy because it's got a mixture of beneficial roles and also negative impacts as well. So, some of the example of the beneficial roles of viruses include its impact in medicine, science, and biotechnology, as well as its ecological impact by recycling nutrients, by regulating populations in the ecosystem. But the bad news is virus causes a wide range of infections in humans, in animals, and in plants. And it can cause economic losses because of crop destruction, livestock epidemics, and it could also be a nightmare in terms of having a global pandemic. So we've come to the end of the lecture, and I hope you'll always remember that viruses are small, they're sneaky, and they're powerful. They shape life in ways that we cannot ignore. So that's it from me. Thank you very much for your kind attention and your commitment, and I look forward to seeing you in my next lecture. Should you have any other questions, or if you want to know more about viruses, please do not hesitate to contact any one of us. Inshallah, all the 5 instructors will be happy and be ready to assist you in any way we can. So thank you very much once again. Assalamualaikum warahmatullahi wabarakatuh. Bye-bye.